Good morning, good afternoon, and good evening, everyone, and welcome to the seventh in this series of the World Cotton Research Conference webinars. And like in the previous webinars, we've got a real special treat for you today. Both our presenters are actually winners of the ICAC Researcher of the Year Award. So we're extremely lucky to be able to get them to participate in this webinar. Let me introduce the first speaker, who is Dr. Mehboob-ur-Rahman, who is the Deputy Chief Scientist and Professor at the National Institute for Biotechnology and Genetic Engineering in Faisalabad, Pakistan. Now, Dr. Rahman works on cotton genome research and translates the genomic information for the development of resilient cotton cultivars. His work has been published extensively in various journals, including 2 in Nature and 1 in Nature Genetics. He has also edited 2 books on cotton. He developed 10 cotton varieties as a principal breeder and took the lead in developing and introducing GM cotton varieties in Pakistan in 2005. Now, Dr. Rahman has received more than a dozen awards, including the ICAC Cotton Researcher of the Year Award in 2014. And we're delighted to have you with us, Dr. Rahman, to talk about genome-assisted breeding in cotton. The floor is yours.
Thank you very much, sir. First of all, good morning and good evening to everyone, and good afternoon. So here in Pakistan, it's rather good evening, I should say, for Pakistani people, scientists. So I am extremely grateful to the organizer of the World Cotton Research Conference for inviting me to make a talk on genomic-assisted breeding in cotton. I will be sharing much more my experience while working with cotton. I'm trying to Can you see my slides?
Yeah, we could see your slides, Dr. Rahman. If you put it onto full screen, then you just need to press the arrow, the down arrow, to move forward.
It is. I tried this, but it's okay now. So okay. Now you can see the slide number 2, right?
Yes, we can. You're clear to go.
Okay. Last year we have witnessed two achievements. That first, the cotton was the first plant that was grown on the surface of the moon by some Chinese scientists, and then the most important, that is, over the last 2-3 years, we have witnessed a lot of papers which are coming on cotton genome sequencing, especially of the species which are not cultivated, and their assembly is very contiguous, and we can use this information for the development of cotton cultivars. So why people they are interested in cotton? Because of it has a lot of important first one is it has it can be the model for, it is a model for domestication as well as the polyploids. We can unravel many genes which are involved in domestication as well as in ploidy level. So the second is that obviously we are getting a natural fiber from this, and it has a lot of scope for the textile industry. And like in Pakistan, India, and many other countries, we are also using its oil for edible purposes. Then finally, the protein that is mostly used in the form of cotton seed cake in Pakistan, India, and many other countries. So it's cotton is for me it's extremely great crop which can be used for by people of different walk of life. So we have witnessed, like from 1970s, I'm not going back, that up to 1990, that the increase in yield, that is from 369 kg per hectare to 574 kg per hectare. So the most important traits, they were the day neutral characters and then early maturity. For example, in our system before 1980, we have the varieties which we cannot, usually the late maturing variety, which usually flowers in November, December, and in January. So now we have the varieties which started, flowering, and the crop get mature in November. So we have a lot of time for cultivating wheat crop. We are having two crops, one cotton, then wheat, then again wheat, cotton, in most of our cotton belt, area in Pakistan. Same is true in many other countries like. So here, as I said earlier, that early maturity and together with some other attributes like resistance to boll weevil, thrips, and diseases. And then we have seen some stagnation between 1990 to 2000, that the, and here you can see that in Pakistan the yield of cotton crop was badly devastated because of the cotton leaf curl virus disease, and we lost 5 million bales. And now, even till now, that every year we are losing around 1 to 2 million bales. If the stress is much more prevalent or much more intensified, then we are like in few previous year, our cotton production that went below 10 million bales. So in 1995, we have seen first GM cotton that was appeared, and we added the genes from the soil bacterium that confer resistance to various insect pests, particularly the bollworm. And the other trait is tolerance to herbicide, and we have seen a jump of 166 kg per hectare between this 2000 to, rather I should say, up to 2015, and then from there again, from 2015 to 2020, again there is stagnation. Every year the world yield is around average 760, 750, like this. So obviously there are challenges which the most significant is the high temperature is the issue in our region. For example, the high temperature, it not only makes the management difficult, especially make many insecticide, they lose their efficacy when the temperature goes beyond 45 degrees Celsius, for example, in our system. The other, it has also synergistic effect on other negatively impacted characters like drought and salinity. So these together, if they appear, they may have potential to reduce the cotton yield by 50 to 67%. So it's really a big challenge, especially for Pakistan, where we are growing our cotton above 45 degrees Celsius. And that's why we have very good germplasm available, which can convert tolerance to heat stress. And then obviously, like many other countries, we are also facing some unique problems like the infestation of pink bollworms, which we have witnessed over the last 5-6 years. And before that, we have the Heliothus and spotted bollworms. And in sucking, we have whitefly thrips. I think these are also these are very common. Pink bollworm, especially, is prevalent in more than 80% of the cotton growing area of the world. So it is really a big challenge to address pink bollworms. And this year we are expecting very low yield in Pakistan, like less than 7 million bale. Usually in the past, you used to have 14 million bales. And in this, the most notorious is pink bollworm, then whitefly, and cotton leaf curl virus. Above all, the other factor, the most significant, is also the high cost of production of cotton. I think in many other country, people, they are too, will be facing this issue. So as I discussed earlier, the cotton leaf curl virus disease is the most significant in our region, and it is, you know, potential threat for the other cotton growing countries where whitefly is prevalent. So this disease should be taken very seriously, and we have developed a lot of improved varieties which confer not absolute resistance, rather high tolerance to cotton leaf curl disease. The other challenges is like as we discuss yield stagnation and low quality. The yield stagnation, the major factor is that yield gap is the major factor between the, you know, for example, uh, in few countries the yield of cotton is very low than that of the other, you know, countries. For example, in Africa, this is around 350 kg per hectare, the land yield, while in few countries it exceeds 1600 kg per hectare. If we exclude the production of India and Africa, then we usually have the world average, you know, is around more than 1000 kg per hectare. And the same is true in Pakistan, that our yield this year we are expecting much low yield, even less than 500 kg lint per hectare. So this is really a big challenge to bridge this gap between the potential yield as well as the actual yield. So then the other factor is that in the past, like breeders, they are trying to develop varieties which can perform better under high input environments. I mean, you apply plenty of fertilizer and everything, and breeder, they usually try to make selections on very, you know, rich soils, very fertile soils. And when especially this issue is very much evident in developing countries, when we grow these varieties at farm field, or the common farmer when grow these varieties, he was unable to, you know, have that particular yield which the breeders they claim, or the breeder they have that particular yield at their farms. So this, and now I think we should all change our breeding strategies as well. So the other obvious factor which is hurting everyone, that is the competition with the, you know, man-made fiber, and it is contributing around more than 60% of the total, you know, fiber demand of our textile industry worldwide. So the other factor is that petroleum prices, they are getting low, and I'm afraid that it may further, you know, compete out the yarn, you know, I mean the lint fiber. So these are a few other issues which, you know, we have witnessed. For example, like growing with GM cotton, and we stop spraying for certain bollworms which indirectly control some bugs. And this millibug that appeared, for example, first time in 2006, and it remained headache up in 2006, 7, 8, and still we find some patches of millibug in the field. So this is another one should keep an eye when we are using new technologies. One should keep an eye that we should not allow the other pests to appear in the field. So we have these problem. Everyone knows about these problem, as we discussed that high temperature, then drought, and bollworms and diseases. They are the real challenge which the breeders are, you know, they are supposed to address. So we can only offer genetic solution. I'm not talking about the managerial management, you know, solutions. I'm talking about the genetic solutions that should be provided to the farmer in the form of seed. As we all know that we have the only when there is an issue that we should explore our genetic window, or we should explore our germplasm, from where we can, you know, derive new genes into our, you know, old or obsolete or old cultivated varieties. And first is the very conventional way, like people they have done in the past, like recombination breedings. Then mutation is another approach that people they use. Radiations are, now we have a lot of publication they are coming using about the use of chemical mutagens, for example EMS, ethyl methyl sulfonate, for adding diversity in the genome. Again, each technology has its own advantage, disadvantage. Then last, the which we GM technologies, which we it definitely it has a lot of advantages when we are using these technologies for the improvement of cotton crop. So once we have discussed what we have and what should be the strategy and methodologies to achieve that particular target. So here again, that we have these 4 approaches available other than recombinant breeding. That genomic assisted breeding, using genomic information for making, especially people, they are making selections by using DNA markers for the trait of interest. And then, as I said earlier, mutation breeding, it also helps in some cases, we have seen. And then GM technology, and now the new technique, which is, you have seen that last year Nobel Prize was given on this particular genome editing technologies. So it helps in achieving precision in breeding, especially the genome editing technology. All others, for example, in GM and as well as in mutation, we don't have control on our on our trait of gene of interest, that we can, like in mutation, we randomly mutate the genome, and then we try our luck if we can find the best mutants. Again, in GM, insertion is again very random, and no one knows where your gene of interest that land on the genome. Then genomic assisted breeding is only to selection in the field. We use selection in the lab using the DNA markers. So all this technology, they are available. For this, we need to have very strong genetic resource available with us. So this is a major weapon of the breeders or geneticists, which they usually use to, either adding new gene, deleting, or editing genome. What you want to do, at least you must have very good genetic resource. So every country, almost major cotton growing countries, they have gene banks where they have a lot of accession in the form of germplasm, cultivar, land races, and even species they have. And most of them, they are in the form of germplasm, and very few, they are land races and some other, And the major issue with this germplasm, that breeders, they usually share in the past their germplasm with their colleagues working in other countries. They usually exchange the germplasm, and I am afraid that most of us, they have duplicated accession. Even in the same gene bank, we have this problem of duplications, right? So look at, they are more than 70,000, and actually to me they will be less than 20,000 if we have a fair representation of the all these genotype. If we avoid duplications, then its number will go very down drastically. So once we have this, that we should know that what our germplasm offer to us for manipulations. So we first, before starting any characterization, either that phenotypic or DNA-based characterization, we must have a complete information of the these accessions. We should avoid duplicated accessions. One, as we, it can be done even in the field. We can have a lot of phenotypic data. We can do that. We should discard duplicated. But there are many accessions which looks very similar, but at DNA level they are very different. So for this, I suggest that we should at least run a couple of markers on those, so that we should shortlist the material for phenotypic characterization at large scale. So when we are prioritizing our genetic material, we should also include all these obsolete cultivars, which have been discarded in the past only because of handicap in one or two traits. For example, in Pakistan, we have very few, we witness very good varieties in the past. For example, CM496 was very promising variety, but it was gone out of field because of the infection of cotton leaf curl disease. All our varieties in the past, they have gone out of the field because of cotton leaf curl virus. Then again 3701, that is in the BT version, again that variety became susceptible to cotton leaf curl virus in Punjab, and that has been gone out of this. These varieties, they have been delisted by our government. So we should consider those cultivar as well. And then lastly, the untapped usually have all these land races and species. We should have a very either we should have very good collections of these for doing this characterization work, and then we should initiate all these projects. So as we know, the people, they in the present time, especially when PBRs they are there, people, breeders, they usually don't share their material. So for those, they should at least screen their genetic material in geographically distinct region within their own country. Or like, it is very advisable if we can have multinational trials, especially across different countries. This germ plants should be exchanged extensively among the breeders, and we should have very good consortium for that, and very good phenotypic platform, like we have done in the past, especially in the case of tackling the cotton leaf curl virus. I will share that particular information in my upcoming slides. So the screening of this material, it should be in hotspot regions for that particular stress. For example, if the cotton leaf curl virus is prevalent in India and Pakistan, the breeders working in other countries, they should screen their material in this region rather than taking all this stuff, the susceptible stuff over in their country, then trying to grow in greenhouse, then screening. It may create mess for them, for those countries as well. So one should be very careful. The cotton leaf curl virus disease is transmitted by whitefly. So in countries like Australia or many other countries where whitefly is prevalent, they should be very careful about this cotton leaf curl virus disease. So they should screen their material in either in Pakistan or in India. Then another, when we are doing these national trials of this germplasm, we should have a very uniform disease rating scale or any rating scale for scoring the traits, so that all breeders or taxonomists, they should be talking the same language. So based on this, that everyone should try to screen their material, then that is ready for screening that is ready for doing some genomic assays. So this is the example of our cotton productivity enhancement that was supported by the government of United States of America, and we shared up to 2018 lot of germplasm, more than 5,000 accessions they were shared, and out of these we identified a couple of accessions which were really either they are highly tolerant or very few they were resistant to cotton leaf curl virus. So from there the USDA department that has the information which lines in their gene bank they are susceptible or which are resistant to cotton leaf curl virus if this particular stress happen to be there. So they have the means to control immediately, rather than like in our system we took 5-6 years to screen the material and developing new cultivars. So this was the we devised this uniform disease rating, very simple, 0, 1, 2, 3 into 4 categories, and we all screen that particular. And if anyone interested, we have a couple of publication on this. They can have the information from that as well. So this is the MAX7. I'm giving you the one example, that MAX7 we identified this. You can see a lot of susceptible plants. For example, these are which are appearing like a cabbage plant, very small. Like in this particular case, also you can see this is a very healthy. In case of 2472-3, then the other one you can see very next to all these lanes accession, they are very susceptible to cotton leaf curl disease. So this one is the maximum 103, the accession number. This was the max 7. In all its surrounding, all these were very, susceptible to cotton leaf curl virus. And this line, performed in all, breeding stations. This line was screened in more than 7-8 locations in Pakistan, and almost all locations it has shown field resistance to cotton leaf curl disease. And it has, but it is not agronomically as good as the other leading cultivars. So it has very small bole and very small leaf, lot of I think gossipwall and hairs on the body. So this was we use Max 7 to improve our cotton cultivars. And here this was information which we retrieved by by running this project, that there are two strains like that first appeared in 1990s. The other recently appeared in 2000, I think, two Burewala virus disease. That again is the same virus strain, but is the same virus but new strain than the Multan or Cochrane virus. So there we have two sources of resistance, LRA and CP15. But here we identify 16 excess. Among those, Max 7 was the most significant. And I use Max 7 extensively in developing, new cultivars. And the major difference between two resistance, the old one was completely dominant, while it has in case of Burewala resistance to Burewala virus, its dominance was incomplete, and it seems to controlled by many genes. For example, 2 or 3 major genes, then there are many genes with minor effect. While the first, resistance that the against the Multan or Cochrane virus, we reported 2 dominant genes, and breeding was very easy for that. So there is a solution. Either we should, we should pyramid both sources of resistance into one genotypes. Then, as I discussed, that we identify more than 16 accessions, but most of them, they were not non-flowering in our system. They were not day neutral. So they usually flower in Mexico or in other countries. So what the solution can be, these should be crossover there. Their F1 should be bring to, in our system. Then we can, that usually flower. Here I suggest, in those days I suggested that, that those we make over there, then we can have a BC1 F1 progenies over there in Mexico. Then we can screen those, genotypes in Pakistan against this particular disease. So now the recent information, what it appeared in one of the publication, which again from our institute, that Burewala virus is no more prevalent in the field. So, and the good thing is this stuff is, we have the stuff now. Let's see what it happens. Again, people, they are reporting the old virus in the field, like Multan or Cochrane. So after 20 years, so again, the old strains, they are appearing up. Everyone can hear me? Hello? Hello?
Yes, we can hear you, Dr. Rahman.
Yeah, we can hear you.
So this is a last month, this was the first variety came out of this project, and that was we use Max 7 into our one of the leading variety 3701, and we use various markers to develop this variety, and within 3-4 years we were able to fix all this, genetic background of 3701 and only the resistance that we derived from Max 7, and it has shown very good yield potential, against the two standards, that is 16% more yield, or for it is the average data of 2 years. In 1 year, it was more than 8%, then next year it was more than 26. So average was around 16%. So this is the advantage of why we should share material, why we should screen material in different, So again, another project we have under Park China project, and this is addressing the heat tolerance. So we have included this trial has been arranged in three locations in Pakistan, and I think in 2 locations in China. It represents 250 genotypes from China, and they have these 250 genotypes. They are mostly, representing different countries, and these are all obsolete varieties or very old germplasm, and we screen those. And we have some very good interesting results of this. We screen for 4 years. So if the temperature goes beyond 45 degrees Celsius, then we have only one genotype left, that is NIBGE 10, that has shown extreme tolerance to heat stress. While if the temperature remains below 45 degrees Celsius, I mean 40 to 45 degrees Celsius, I'm talking about data, and it is the field data, not the lab data or greenhouse data. So there, we have a lot of stuff, which have been evolved in different countries that has a lot of tolerance against heat. So the big issue with the papers they are coming up, that most of the molecular biologists or breeders, they usually screen their material at less than 40 degrees Celsius, and they usually do seedling experiments. And I think we should have the complete yield data while growing these plants in the field, because they are supposed to tolerate that particular stress in the field. If there are many other stresses there, if heat is there, then you should expect many other stresses. They will also make the plant more unhealthy. For example, when there is a heat, then automatically drought usually appears. So the plant usually lost their competitive ability also, in the field. So I have shown a lot of everyone knows that the shedding is the very common phenomenon. Shedding of immature boll are scarce, and we rated this material at 4 categories, and highly tolerant, then tolerant, moderately subtle, sensitive, and then highly sensitive material. So a lot of genotypes, they cannot survive beyond 45 degrees Celsius, and the night temperature was around 29, 30 degrees. It is more important, night temperature. I missed that, right? So next, once we have very good screening, and all this information I shared with you because that everyone is must be facing such problem in their countries, and what we face. And then once we have this, then then you can develop very good populations on that for mapping or for identification of DNA markers, or doing many others for isolating genes, and you can do a lot of things. The conventional linking mapping experiments, as all we know, that mostly the two parents, they are involved in developing populations. And these are the F2 recombinant inbred lines, the near isogenic lines. They are usually derived from backcross population. So in this particular case, most of the mapping experiments, I think more than 80%, the populations were developed by attempting interspecific cross. I mean cross between 2 species. Those are Gossypium hirsutum and Gossypium barbidans. So when we use these 2 species, then these species, their genome, they have their own dynamics, as we learned. They don't recombine very frequently, when we cross hirsutum into hirsutum, we expect a lot of recombination. In that particular case, we have, at the end of the day, we end up with the hirsutum chromosomes, then in few populations we end up with the barbidans chromosomes. So this is a very major drawback using this interspecific population, and that's why I hardly seen that the markers, such markers which are identified using such populations, they were used in developing new cultivars. So then, to avoid this issue of and usually biparental populations, they have, as you know, that they have low genetic, low resolutions, and we don't have enough, we cannot detect a lot of polymorphism in the genome because only two parents they are involved. And the marker identified by using such population, they are very specific to particular populations. So the next day, to overcome this issue, the genome-wide association mapping strategies, where you can use a lot of accession, at least you must have more than 200 accessions, genotypes, which should be scored for any particular traits in the field. Then you should, then we must have the genotypic information, I mean the information at DNA level of all these accessions, then followed by we can, associate that particular diversity with the phenotypic diversity to identify very stable QTLs and the associated markers. They may have some worth for using in breeding. So these are different methods. Everyone knows these are first two, they are very conventional. Then people, they are using GWAS, then TESEL, and the two model, mostly the mixed linear model people they are using to have the validity of their DNA markers with our QTLs which they are identifying. I have seen couple of papers which are really very good. They have used multi-parents. I mean, they identify diverse parent more than 8. Then they making cross A with B, then C with D, then E with F, and G with H. Then next year, then AB with CD. In this way, they put all the genetic recombination in one F1. Then they develop population out of that up to F4, 5, 6 for doing such experiments. So, as I said, that once we have the very good data, we have very good phenotypic data, and we have very good populations, then we can use the DNA fingerprinting assays. Either the first one, they are very conventional. Now, people, they have extensive use RFLP for estimation of genetic diversity, and very few markers, which have association with some qualitative traits, like we identified with the resistance against first Cochrane virus, 1 or 2 markers. Again, the marker, they are not very close enough to the gene of interest. So they have an issue that we cannot explore all the variation in the genome. Then again, SSRs, now we have a lot of SSRs available. I think with the given genomic information, we have more than 70,000 SSRs. They are available, which can be used in genome-wide studies, especially SSR. Still, I feel very good for the scientists which have not enough resources. They can use these markers very comfortably. Otherwise, other markers which are aiming for identifying SNPs, single nucleotide polymorphism, they require a little bit more investment compared to this. Moreover, these SSR, they are much more, informative as well as reproducible, and they can be automated. And we have seen a lot of, machines. They can, help in, fractionating these SSRs, these SSR fragments, in their own system. So in SSR, again, EST SSR, they are much more preferred because they are directly coming from the major gene, I mean the active part of the genes. However, again, their number is limited. So among SNPs, now everyone is talking about resequencing. 5 years before, GBS was very popular. Then now cost of sequencing has been gone down significantly. The people, they are resequencing the germplasm or accessions to identify SNPs, and followed by then associating with those SNPs with the phenotypic diversity. There are a couple of SNP chip available, and this information has been published by two research groups. One is 63K, the other is 80K. This also equally helpful if we want to identify SNPs among the various genotypes. So then, as you know, there are very well automated system available in the world, especially in advanced countries. They can, in limited time, they can have a lot of genetic resources, a lot of information. Then again, the only issue with these that they have the challenging task of identifying SNPs in polyploid, like cotton, the cultivated tetraploids. You know that it has a lot of copies and a lot of duplicated copies of the gene in different chromosomes. So it is very difficult to assign that from where the SNP is coming. So I have seen this. I have gathered this data from Cotton Gen website, and on April 1st, 2021. So obviously there will be many other QTLs which are not reported in this particular database. So there are around more than nearly 7,000 QTLs they have been reported, and most of them they are aiming for quality traits. Among quality, fiber length, strength, elongation, fineness, Mike, they are identified QTL for these particular traits. So for yield, they were around 1,300, one trait, stature 130, then phenology 2-1, then again addressing the morphology and physiology, you can see on this particular slide. Then for biotics, it is 273, and most of the QTLs you can see here, they're for verticillium, they're around 215, and very few they are for bacterial bolides. So for abiotic stresses, I cannot find these abiotics, QTLs for abiotic from this particular database. I was editing a chapter, so from there I got this information that for abiotic stresses, particularly drought and salinity, people they reported around 96 QTLs, and for high temperature, especially the data from people they gathered data from field and then identifying QTLs, that is also missing. That is not also available. So you can see that most of these QTLs or most of the associated markers with QTL, they have very little worth in terms of cotton breeding. Very few people, they are really a good stable QTLs which can be used in improvement of cotton. So for example, as I said earlier, that people they usually do lab experiments for identifying the QTL associated with complex trait, especially for abiotic stresses. So in the field, when we apply that information generated in the lab, in the field experiment, it is very difficult to translate that information. So again, you can see a lot of biochemical. Again, people they identify such QTLs by doing some biochemical testing, and the yield-related traits they are again not very stable. I mean, they are most of them they are population specific. So now I think now we have a lot of genome sequence information of a lot of species available. I am sure in future we will have a very interesting time. We can unravel or we can identify all these genetic loci or the genes which we can play at our own will. So another is that here I can quote few SNPs which are associated SNPs and associated QTLs. You can see in first, there are few. Obviously, there will be much more, but I only, I'm quoting very few. Like you can see the most latest. I can find 2018, 165 QTLs. They have been identified, which confer resistance to drought and QTL. So similarly, For drought tolerance breeders, they had this scientists, they use the population, again interspecific population, hirsutum and utomantosum, then make the BC2F2 population, and they don't have shown the QTLs, rather they only show the SNPs they identified. Again, the paper appeared last year by Yuan et al. In 2019. These SNPs, they are related to salinity tolerance. Again, most of these SNPs, I think they are coming from lab experiment. Yes, I have seen, I'm also the part of one of paper by one of our Chinese colleague, Dr. Zhang, Professor Zhang, and again, all of us, we prefer to screen the material in the greenhouses at seedling stage, especially for this very complex stage, which, for drought, again, for salinity, I have seen, for wilt, I have seen those experiments, they inoculate in the green. Yes, we, for doing having primary result, we can screen in the greenhouse. Then we should try to correlate or translate those particular results in the field as well. Either they are transferable. If they are really transferable, then we can use the information for an improvement of cotton. Otherwise, we will have a lot of publications. So again, you can see this is the Jiva stuff. Again, lot of SNPs, people they identified, and most recently I have seen this one is again 20 QTL for drought and 23 for salt tolerance, and the gentleman identified, I think.
Dr. Rahman, we will have to leave at least 10 minutes for the Q&A. So here at 8:51. Yeah.
Okay.
Thanks. Thank you.
So these are some, again, quality traits. You can see the key message is that for all quality traits, first, they are distributed throughout almost all chromosomes. And secondly, that in few chromosomes, their number is very high than that of the others. But again, it is very difficult to find very significant QTLs, which can be really used for the improvement of cotton. As I said earlier, that 80% maps, they have been built upon using interspecific populations, just to have a lot of diversity in the populations, to identify more, polymorphism at DNA level. However, as I said earlier, their utility is not as good enough as what we were expecting. So we should, like now, again, the most important message is stable QTLs with maximum phenotypic variance. That is required for us, for breeders rather, I should say. So in marker resistant selections, as we were discussing all these access for doing the genomic assisted breeding or marker assisted breeding, then again, very simple, this slide everyone has seen, that we usually have two parents, then F2, and F2 usually makes selection using that particular DNA, and very few plants we usually select to advance that particular generation. So here, is the message, especially, for complex trait, like in our case, we did a lot of work on Budewala virus, and here we identify these particular markers. So here the plants which are first 6 plants, they were rated at 1. I mean, very low disease intensity, like they were tolerant. Then the plants 7 to 14, they were rated at 2. I mean, they are susceptible. The next group is highly susceptible. So if we have these markers, I rated 1, they're coming from the resistant parent, a resistant parent like MAX7. If we have this allele, 5 and at least 4 alleles which are coming from the resistant parent, they usually confer very good resistance in the field. All others, if we lose 2 alleles coming from the resistant parent, then we don't expect a very good tolerance in the field against that particular stress, especially I'm talking about cotton leaf curl disease. So then backcrossing people, they hear the message is that we should use both foreground as well as background selections for the identification of our plant. And for complex trait, we should also correlate this selection, actually what they are doing phenotypically. All this information together, we can have the best recombinant. And this method is very useful for when we are using different species in our breeding program, especially in introgression breedings. Look, I'm skipping this. So these are the list of traits which we can introgress. Rather the people, they like bacterial blight from arboreum. Again, arboreum is a very good source for building resistance to cotton leaf curl virus. Then fusarium wilt from Gossypium australe, then rust resist, then verticillium from again australe. Then so this we have done this where we have developed, rather introgress the monitor the introgress part of G arboreum. We identify these markers, and these markers have a very good relationship with this cotton leaf resistance to cotton leaf curl virus. We crossed arboreum with the hirsutum, and then we introgress the alleles coming from arboreum in our material. So the mutation everyone knows, and EMS I found is very good. As you can see, a lot of variation we have. Only one message is that we should identify if the real man-made mutations are they are natural available in the population. As cotton is often cross-pollinated, and breeders, especially working using various mutagens, they are very happy that we find a lot of variation. Actually, these are natural variation, not the induced mutations. So we should be very careful for this. And you can see that one can also use this. So these are we should try to make libraries and with very good photograph. And these mutations can be, we usually exam capture assay in case of wheat. And if we have enough resources, you can resequence these mutants, then validate. Validation is very much important step.
Dr. Rahman?
Yes.
Is it possible to conclude now, because we're running out of time now?
Okay, I can go I can go to the if I can have 2 or 3 more minutes. Is it
okay? We're eating into the question time, so yes. So please please conclude.
This is the okay. So this is then we have sequence information. Then genome editing is, everyone is using this technology for editing the genome. For this, I will discuss some limitations of this. For example, we must have the complete information of especially the complex genetic pathways, so that we should know our targets. This number one. And number two is, in case of cotton, a transformation is still required for these cassettes to like you are using coker, and first we are supposed to transform that particular cassette in the coker, then edit those, then we should through back cross breeding or through breeding again into our cultivator. So this is the major limitation in case of cotton. And lastly, we are using, most of the time we are silencing the genes. So yes, it's very good if we have some genes which are involved in some negative characters. Yes, we can silence, but to enhance the expression is still very challenging. I have very few reports I have seen that they have improved resistance. Only 1 or 2 reports in case of cotton they are available. So these are the advantages over our conventional system versus this genome breeding. Again, if we can edit the chloroplast in our system, we are doing a lot of work on chloroplast transformation, but still no product came out of this. It has a lot of info, and the future is very golden for cotton genome-assisted breeding, because we have a lot of sequencing informations available for all, including all progenitor species. Hopefully, we have the platinum standard genome sequence information available. and that can be used especially to target the rare alleles. So this is next is that we should have, for doing this precision breeding, we should have very complete information. For example, before doing GWAS, we must have very good phenotypic characterization using various phenomic platforms. Then consortium-based science should, I will again, then gapless genomes and pangenome is required, as we have witnessed in case of rice. So again, if we can transfer some genes from some very drought-tolerant species into, like this Procera, into cotton, this was really in Pakistan and India to grow on dry land and saline environment. I'm sure it must be growing in some other countries. We can have very good tolerance to drought, heat, and many others. Thank you, Hermes, for nice listening.
Thank you, Dr. Rahman, for a very comprehensive presentation. I'm really sorry that we've run out of time. And it's just perhaps it's indicative of how much interest your presentation has stirred is the number of questions in the question and answer chat box. So over 20 questions. Unfortunately, we're not going to be able to have time to answer those questions now. So what I'm going to suggest is that we proceed with the next presenter. And Dr. Rahman, if you could look in the Q&A boxes and perhaps answer some of those questions. And if we've got time, we can come back to those questions at the end, because there's some really interesting questions in there. And I think that we would certainly like to hear your opinion on some of those. But if you could have a quick look through and see which ones you can answer, that would help us filter out some before the end. So thank you very much indeed, and we'll come back to you later on. And now it's my pleasure to introduce our next presenter, Dr. John Yu, who works as a research geneticist at the USDA research facility, and is also an adjunct professor at Texas A&M University. Dr. Yu has about 4 decades research experience at national and international institutions and is globally renowned for his outstanding work on plant breeding, genetics, and genomics. He has authored more than 100 peer-reviewed journal articles and other publications with 10,310 Google Scholar citations. He's mentored more than 60 students and visiting scientists from the United States and abroad, and he's played a seminal role in cotton genome mapping and sequencing, including the public release of portable genetic, physical, and genome maps of core cotton species. Twice selected by his global peers to lead as the chairman, Dr. Yu advocates for the International Cotton Genome Initiative, ICGI, in order to increase knowledge and resources of cotton genomes for the benefit of mankind. He also serves as the expert member of the USDA National Life Science Patent Committee, amongst many other scientific advisory roles. And I think that's a real good intro to you, Dr. Yu. But finally, we mustn't forget that the most important, of course, is that you were the ICAC Cotton Researcher of the Year for 2019. The virtual floor is yours, Dr. Yu.
Thank you, Kay, and Kishar, for your invitation and the introduction to this webinar lecture. It is my honor to present and discuss the power of cotton genomics to genetic improvement. The previous lecture given by Dr. Mehboob-ur-Rahman today and by Jonathan Wendell 2 months ago already showed the power of the cotton genomics. Prior to my arrival at this USDA location, Texas A&M University at campus in 1994. I had worked on rice at the International Rice Research Institute, Cornell University, and other institutions. I much appreciated the opportunity and the challenges of Gossiping genus that is the source of the world-leading natural fiber and an important oil seed in the global market. I became interested in understanding the genetic variation in cotton plants through integrated mapping and sequencing of the larger complex genomes, as well as through molecular analysis of genetic diversity, otherwise buried in the gossiping germplasm. Does everybody see my slides fully?
Yes.
Yes, we can. Yes.
Okay, I just double check, make sure the, hold on, let me see. And looks like I do a little bit of adjustment here. All right. Just bear with me just a second. On this campus, the Texas A&M University campus, we have a USDA location. The slides here showing the bird view of the location, the lab here in building 1 and 11, and also seawall. We have a cotton working collection here in culture station, and a greenhouse here, and to grow the maintain the wild species. So in the collection, we have about 10,000 accessions represent all the 50 species of gossiping genus. But the value of the collection has been barely explored because of the lack of a good descriptor. And this hindered the effective characterization. So identification of the beneficial gene and the QTL and the regulatory network in cotton genome and the subgenome or individual chromosome would shed light into the evolutionary and functional role of that played at the various stages of the cotton fiber and seed development. And in the latest 20s, Dr. Esbrook and Brauman had a pretty good review, very nice review, and indicating the genetic base of the cultivated cotton become very narrow, and a decline in the cotton yield quality occurred. And this is because of two main reasons. One is a lack of sufficient effective tool to facilitate genetic discovery, gene discovery in the germplasm, and transfer this gene into the elite lines. And also another reason, some major biotechnological initiative in cotton will focus on transgenic to protect the cotton plants from the insects and other disease. So the erosion of the genetic base and the decline in the yield and quality concerns cotton breeders. So in the late 90s, the geneticist and the breeder from a few research labs began to develop different types of molecular markers and to map them in cotton genome for marker-assisted selections. And this is types of markers initiated in the 1990s. We had Dr. Cole and I had a review on the status of this marker early days, and only the other marker type SNP was really after this early mark types. And because of this situation, a small group of cotton research organized a symposium, international symposium on the molecular markers in Australia in early 2000. And the International Cotton Genome Initiative was conceived at this meeting, but formalized with the first workshop in France, which organized by Marc Gibbon, who is here in audience. And he and other early advocate for this international collaboration. So the mission was set to increase knowledge of the structure and the function of the cotton genome to benefit the global community, and to facilitate global communication, collaboration, education, and integrated knowledge, develop technology, resources, and plan coordinated research. Currently, around 500 ICGI members from about 30 countries. And shortly after the Australia meeting, the US President Bill Clinton and UK Prime Minister John Major jointly announced the completion of the first draft human genomes through the 13 years international collaboration. And this news published in Nature and Science and other media really inspired the ICGI and as well as other plant animal research community to seize the opportunity of so-called genome revolution. And we organized, immediately organized another workshop in Nanjing, China, and that would receive individual research presentation and established a 5 work group to coordinate the structural functional comparative genetic stocks and the bioinformatics aspects of the cotton genomics research. And the ICGI has been expanded rapidly in research collaboration and coordination over the past 2 decades. Since 2004, in Hyderabad, we have been holding the biennial conference, research conference in India, Brazil, China, and the US, Australia, and Scotland. And due to current pandemic, the conference to be held in Israel last year was postponed. And meanwhile, many bilateral collaboration were initiated, coordinated in cotton genome research. This photo was taken in at the 2005 workshop sponsored by USDA and Uzbek Academy of Science in Tashkent, and hosted by Ibrahim and his colleagues. And a substantial mapping of the cotton gene and the molecular characterization of the cotton germplasm was accomplished. And in 2007, a collaborative agreement to sequence the upland cotton genome was signed in College Station, Texas, between my former research unit leader, Dr. Russell Cole, and the former director of the China National Cotton Research Institute, Dr. Xuxian Yu. And we agreed to a sequence of text marker number 1, because that's a genetic standard of the upland cotton. And following the publication of the white species Gossipin in 2012, the reference genome of the cultivated diploid tetraploid species Gossipin arbora and Gossipin hirsuta TM1 were completed in 2014 and 2015 respectively. And this groundbreaking report laid the essential foundation for the remaining cotton species and many thousands cotton accession around the world. And since then, more than a dozen cotton species, including all cultivated cotton, I highlight here in bold, and all tetraploid species AD 1 through 5 have been fully sequenced. And also many thousands, the germplasm accession was sequenced. So the information, the abundant sequence information provide a very good opportunity for genetic improvement. And thus far, the ICGI community, research community has accomplished the millions portable cotton SSR and SNP markers. That's the original goal when the Australia meeting was held. Needed lots of portable markers. So that's the SSR and SNP type of marker. The community feel they are the best type of marker for the breeding purpose. And also the thousands QTL was identified for cotton traits, and also thousands genes coding the protein annotated, and hundreds of genetic physical maps, and also dozens cotton genome sequence I just mentioned, and also another centralized database with bioinformatics tool was developed. That's in collaboration with Cotton Incorporated and Washington State University and the broader cotton research community. And I'm switch the gear a little bit to describe how we reach this accomplishment. The approach we're taking. As we know, the genetics is a study of the single gene in the organism. And the genomics is a study of all genes in each genome of an organism. And cotton genomics is a powerful tool to study all the cotton genes in each genome, or all the genome of the species, the 50+ species. So the next set of slides, I'm describe the approach we take, not only our group, but other community also included. By the way, is everybody hear from me clearly, the voice and the slides?
Yes, yes.
Thank you. Thanks. And at the very beginning of this century, cotton was much behind other major crops plants in developing genomic resources. And we identified a critical lack of the integrated genome map that would serve as a raw map for cotton genetic research. So this is like a backbone, the high freeway. If we needed to have a very good infrastructure for the genetic improvement, we needed this, and the same as similar to other crop communities. So this would have integrated genome map would have the precise and rapid mapping of all genes, and we understand the genome organization and gene expression, and also the mapping all the markers, and so we can manipulate the traits, the genes with marker-based analysis. And also the functionally, we need to understand how the gene regulated and expressed and their interaction. So this infrastructure, we try to build using this framework, or called the first, the chromosome is a piece of DNA. It's a very long stretch of DNA. And with centromere, some were identified on the chromosome, but the physical map we just resemble this chromosome with a piece of the fragment, the DNA. And this is each fragment or contig, they represent a certain size. And so in a million base pair. And the linkage map, we using traditionally the two parents, biparent, or some later on the other methods to find the marker in the recombinational way of the distance. And cytologically, we need to identify the marker and the chromosome, the landmark zone at each position, the physical position. And then transcriptionally, we need to identify the gene or exon or express sequence tag in each location. Then finally, we need to get it down to the DNA sequence space level. So this is the idea we we used for the integrated genome mapping, and using the SSR marker and the back fingerprints, and also EST unity. And now we replace them with the SNP markers and the sequence context and the annotated gene. So that would advance to the next level. And the first I like to describe why we study the upland cotton as our the initial goal, because the upland cotton has an allotechopod genome. That's very complicated, and because lots of duplication, and there are 26 haploid chromosomes, and the genome size is about 2.3 billion base pair, and genetically about 3,500 centimorgan. And now we know that there are 75,000 genes approximately, but many of them are duplicates because the tetraploid, they have 2 copies at least, and some duplication segment, they have more than 2 copies. And the most important, they are cultivated over 90% in the world. And with the support from the cotton research community, we choose the choose the text marker number 1, because many genetic and the cytogenetic stocks, including mapping population, have been developed in this background. And Dr. Russell, my former unit leader, he had a long history doing prepare this genetic material, and actually purified this line with 40 or 50 generation of the control cell. So this the seeds are very pure, very complete homozygosity. That's help our sequencing. Otherwise, it would be like a human complete some heterozygous issue we need to resolve. And then we constructed 3 libraries, and with the back library. So it's a bacterial artificial chromosome library, and using the different enzyme, restriction enzyme. This is Hindi3 enzymes, and the other 2 library are ECO1 and BAMH1 restriction enzymes. So the insert size, the fragment size of cotton DNA is was about 150 kb, and clone into the vector, so the DNA can be fingerprinted at that time, because the sequencing cost was still high. And as we know, the cotton community, the funding was a major constraint. And we fingerprinted each clones, and about 100,000 clones we fingerprinted. And then we assembled them with the fingerprints into the contig. And then we using this contig to build a physical map. And this is the machine we early on used for the fragment sequence analysis. And this is capillary-based, but some marker, SSR marker, we used gel-based also at that time. But quite a few, the sequencing tag and back-end sequence use this machine. and even the SSR dye-labeled using this capillary system. And last week I had to retire this 3 such a machine because the technology advanced. Now the high throughput will be make the DNA analysis more efficient. So this is all the slides, all the picture of the fingerprints look like, and this is the control, the markers, and in between the cotton DNA, they have different fragments of fingerprinted. And then we developed the SSR marker from the backend sequence. And this is from the backend, the sequence the backends. And about 60% of the marker, we find that they produce a single band. So that means there can be a locus specific or used to anchor the subgenome of the back context. And the first 2 the mapping populations, so we also put them into the genetic map in this population. And this is a mapping population derived from the cross between TN1 and 379. 379 is the genetic standard of biodensity. And because the interspecific polymorphism was low, and we initially used the interspecific, as Dr. Rahman mentioned, that's one challenge to the cotton, the polymorphism rate at the early stage. And also we grew this segregate the mapping population in the field in a different location, the diverse location environments, and the plants germinated in the greenhouse and transplanted into the field, and then we do the each for the field application observation, segregating traits, and then we constructed a genetic linkage map, and of all 26 chromosomes, here is showing the 2 pairs of chromosomes, and chromosome 2 and 3 and 14 and 17, they showing the partial homology and also translocation between. So there was some genetic variation and involved in the structure we find, and the other 22 chromosomes showing some kind of duplication. I didn't show that. And also we put the traits, the cotton fiber and the yield and other agronomic traits into the map based on the marker linkages. And then we had the overall hybridization of the EST. Cotton EST library was built by the community, and we had used this to design the overgrowth, the 2D or 3D overgrowth probe. So they double spotted on a high density TN1 back filters, and this double spot showing the position of the EST gene on the back clone. So that serves integration to the map. And so this is one fragment, one conjugate of this physical map with linkage of the genetic marker and also EST associations. And this is one chromosome of the integrated genome map. In the middle showing in the center showing the genetic distance of the markers, mostly SSR and some SNP marker already starting. And on the left is the gene, the EST unit genes, and on the right is the physical conjugate from the back clones. And also I need to mention when we do the TN1, and we have to think of the 2 subgenomes resembling the probable progenitor of the diploid cotton. So we used the Gossypium arboreum and the Gossypium remondia to be a presumed progenitor to the diploid, to the tetraploid subgenomes. So we had to sequence these 2 before we could complete the TN1 genome. So this is in 2012 we reported completion of the genome, the 13 chromosomes, and assembled, and the green showing the gene density along the chromosomes and some other elements marked here on the chromosome. And a couple of years later, we sequenced a bodium that's another subgenome A, and we find the very close relationship with the cacao. So the cacao, the genome was already sequenced, so the genetic information, genomic information could be used for cotton, and and also we know that the genome size between the A and the B is about 2:1. And this is a gossiping bodium, and this is a gossiping Romandia. And this is a small scaffold of this chromosome number 7. And the actual 2 million base pair, this is the 1.5 million base pair. And this is a 3.5 million pair. And the difference we see is mostly based due to the transposable elements. And the number of genes pretty close, 191 and 216. But the repetitive DNA is more than twice of the D genomes. So that's the A genome expansion due to the repeats. And also this panel D showing the genomic variation among the genetic synthetic block, while some minor the minor variation observed between the D and the DT, the diploid D genome and the subgenome D. And major changes were observed among the 3 A genomes, the Arborium and the Herbaceo and the TN1AT genomes, and including 3 reciprocal translocation and 2 larger inversion here on chromosome 10 and 12. And this figure illustrates the speciation of the diploids A1 and A2 Herbaceo and Arborium involved independently after the formation of the tetraploid AD upland cotton. And neither diploids A1 or A2 was the true donor to the AT subgenome. So all existing A genome, including AT and A1, A2, originally from these unknown ancestors. So the clarification of the A genome origin helped our understanding of fiber development in the upland cotton. And the cotton fiber development has 4 distinct but overlapping stages of the fiber initiation, elongation, second cell wall deposition, fiber maturation. And this stage of the development controlled by almost half of the genome. The fiber transcriptome was really involved with about half of size of the genomes, as we estimate. And the one thing we were curious about how the union of the 2 diploid cotton, one produced a short fiber, the A cotton, and another produced a non-spinable fiber, the Y species of Romandia. When the 2 hybridized into the 1 nucleus and produced much better the cotton. So to answer this question, we analyzed the whole genome. And since we noticed the regulation of the ethylene, the compound. So for the ethylene is necessary for the fiber initiation and elongation. This is a tetraploid upland cotton. And but either way, if 2 too little, like this line, the arboreal, and they will suppress the fiber elongation. But if too much overwhelmment shuts down the fiber elongation, so this arboreal, the Romondia does not produce spinal fiber. So this is one contributing factor. And we need to balance out with the two in the same genome, then make the ideal the tetraploid upland cotton. And then the fiber gene in the but at these 4 stages, we also structurally located on the chromosome, and we find the gene island, the major gene island for fiber initiation on chromosome 5, and also elongation on chromosome 10 and 14. And the second cell wall deposition and maturation on the 15. So other chromosome, they have the gene, but this predominantly we have island identified. And the study we in our lab, we identified about 500 contexts related to fiber genes, and then assembled them to 150 million base physical map, and about 6.7% of genome size. And this anchored the individual chromosome and the fiber development gene were more from A subgenome than D. But interestingly, the D contributing to the fiber by the way to regulate them. So the transcription factor, we find more from D subgenome than A. So we have more fiber gene from AT and more fiber, more transcription factor from the DT genome. The interaction between 2 subgenomes in one nucleus really enhance the fiber development production. So this, to some extent, answer our question, how the 2 diploids which produce short fiber, no fiber, would in combination produce longer fibers. And all this, the gene responsible for various stages of the fiber development were physically located on cotton genome sequence map. So we have this context, we just learned them and identify them into the different locations. And next few slides, I'd like to specifically mention the application of the in collaboration with University of North Texas, we identified in the collection naturally occurred variant for improved fatty acid profile. So this is the DNA fragment we identified in this germplasm and enhanced the cotton oil seed oil, cotton seed oil. And the U.S. National Cotton Germplasm Collection has other accession we screened, but this particular line stand out. We could use it for the cotton seed improvement because the high oleic seed oil have a metal price premium, increase the market share for other oil seed crops. And this represents a valuable association for cotton breeder to examine more closely. And also the in collaboration with the Uzbekistan scientists, we put constructed the map, including the phytochrome for the photo period sensitivities. So this information will be used for the exporting the exotic cotton germplasm that otherwise do not flower in a normal condition. So we can integrate this information for the cotton improvement or germplasm enhancement for elite lines. And also in collaboration with Chinese researchers, a whole genome selective sweep analysis among the relatively breeding pedigree line showing the gene for biotic and abiotic tolerance. And this is a fungal disease. We find that scanned the 26 chromosomes showing the some very interesting, very strong signal for this fungal disease. And also we identified a similar genes for the salt and the drought tolerance. And this results are very useful in the breeding for this biotic and antibiotic tolerance lines. And also the in collaboration with Texas A&M University, we the cotton susceptible gene was silenced. We used the virus-induced gene silence to knock out the susceptible genes, and so they can have normal growth after the infection. And this will the discovery will facilitate strategic development of the durable and broader spectrum resistance to disease in cotton production. And so as mentioned before, the cotton gene is the community database. And this database, we collaborated with Cotton Incorporated and Washington State University and many cotton researchers around the world, and especially in the US. And the information is very rich. We have over the last dozen years, so more than 500,000 markers and 6,500 QTL and 25,000 millions genotype data, and more than 500,000 phenotypic data are curated in this, and also 45,000 images for on the germplasm also here in the database, and the database including breeding information, genetic information, and the genomic information. So it's accessed freely by the cotton researchers around the world. And since 2016, the database was used from 88 countries to 114 countries, and the 2,000 about the 3,000 user increased to the almost 6,000 users. The time, I mean, not some user used more than one time, so it's not just a person, it's the users. And lastly, before I conclude my talk, I like to go back to the genetic diversity, briefly discuss how we take advantage of the gene bank, the collections around the world, including the US. We initially developed this 105 core SS markers, and these core SS markers are of high quality and well characterized, mapped along the chromosome. So each chromosome arm, we selected 4 markers or 2 markers. And the reason we use the 105 because the chromosome 5, we choose the 5 markers and the 3 marker on the long arm. So that way we can triplex, multiplex with the dye label, and for the PCR, capillary-based PCR analysis. So these sets of markers were used to characterize the US collection and other collections around the world. And so in the US collections, we find the upland cotton and the Hisuian and the barbadense and other species, they have a very rich cluster, the diversity, genetic diversity. But there are some exceptions we can see it's scattered around that we're showing some interesting integration. So the integrated species accessions from one another because this is the two species, upland cotton and Gossypium herbaceae, and this is Gossypium barbadense. And the number of the accession, they're showing at least 10% of integration from other species. So this is the information we like to understand more for the breeding purpose. So the yield can be increased for the barbadense, but the fiber can be also incorporated into upland cotton, the quality genes. And more detailed survey from these collections we are doing is being done with a snipper chip. And as Dr. The chip, there are two chips. We use the 63K chip and to characterize this, and eventually we like to have individual accession sequence that, for the sequencing scan purpose. And this graph was is from the U.S. National Human Genome Research Institute. And with DNA sequencing technology advanced rapidly over the past 2 decades, from 2001 to 2020, the cost to decode the complete genome of a species reduced dramatically. You see that exponentially reduced. And at the early stage, the first human genome, I checked some history last week. It cost 3 billion and 13 years to sequence the human genome. And this is 2001 would cost 100 million. And of course, nowadays we can do this one genome for $1,000, even under. And but the reference genome and the bioinformatics tool to analyze the genome would help much of this cotton research. And cotton is unique and different from human genome in terms of cotton has more than 70% of replicated DNA, and the human genome, of course, heterologous. But the cotton genome has lots of duplication and all the tetraploid, the 2 copy and the additional segmental duplications. So the each species has different challenges, but with technology advancement, we could use this genome advancement to speed up the cotton research, genetic improvement. So now we are exploring this genetic diversity of the cotton collection with individual accession being sequenced and resequenced since we have reference genome. And making the cotton germplasm truly the gene bank. Sometimes we call the germplasm bank collection as a gene bank, but the next stage really we will categorize not by accession but by the gene and each accession. So the breeder can use this information just like a library and like a bank. You can get this for a particular trait and phenotypic improvement. So the post-genomic application from the lab bench to the farmland in the foreseeable future will be possible with multidisciplinary approach to organize, synthesize, browse, and translate in a newly derived knowledge base of genetic variation for enhanced cultivar development. And the recent advance of cotton genomics is moving cotton breeding into a new era where molecular genetics will focus on the functional trait for improved cotton cultivars. And with that, I like to thank many collaborators and supporters to the cotton genome research. Without the global collaboration and coordination, it would not have been possible to achieve the progress. In the same spirit, the full potential of the cotton genomics to genetic improvement will only be reached if we continue to work together. And besides the ICGI, I appreciate the critical role the ICAC and the Cotton Incorporated are taking. And with that, I'd like to enjoy any question, and we can discuss it with the next 10 minutes.
Well, thank you, Dr. Yu, and I have to say thank you, Dr. Rahman, for answering all your questions and even having a go at Dr. Yu's questions as well. But we're not going to let you get away with it, Dr. Yu. You're going to still have to answer some of those questions.
Yeah, I have 10 minutes.
So the first one, I'm going to try and find some from Dr. Raghavendra. Though thousands of QTL have been mapped, what is that there? Why is that, sorry, that there are hardly any studies about successful mass and QTL integration, especially with respect to cultivar development and genetic enhancement? Did you understand that question?
Yes, yes. So this is a question that people often ask. This is an excellent question and very persistent. It's being asked many times. And as we know, the QTL analysis, it's just estimate. It's a statistical analysis, and there are lots of factors. It's computer analysis. So the statistically significant QTL quantitative trait loci, but you don't know how far from the real gene, because we know the most of the traits, the plant traits like cotton, controlled by the polygenic inheritance. So the gene, they scattered around the genome. So the QTL sometimes detect part of this network, but not all. And in some cases, if you're missing critical elements, the trait will not be altered or improved. I mean, they needed to have a whole system. So that's why it's very frustrated, not only in cotton, but other crop species, even in the human system. The QTL analysis is a rough estimation. So we needed to have a higher resolution genome map like a sequencing. So we really understand where those multiple loci polygenic traits are located and how they interact. That's the most important thing. So the community estimated if you have a 10 to 20% of the QTL showing effect in the breeding program, that's already excellent. So so that means 80% or even 90% of the QTL, you may not be able to see the real effect in the breeding program. And that's the problem of the traditional QTL analysis. We need to have the genome sequencing data and most advanced biological knowledge base.
Good, thank you. I'm not sure if this one was for Dr. Rahman or it was for you, but it's an interesting question anyway. It's from Dr. Raj Kumar. How many markers do you suggest for background selection for ideal RPG, recurrent parent genome recovery?
Of course, the marker, the number, the higher the better, and because it will affect the resolution. But if you try to recover the recurrent parents from the breeding program, and we usually like to have at least several hundred markers, and at least you need to use those to show whether the background genotype represented.
Thank you. And from Ashok J. Bai, what is the process to identify homogeneous markers, SNPs, in cotton?
The cotton is a very complicated genome. You have the two subgenome, and they are partially homologous, and also the segmental duplication. So we need to identify the three types. One is heterozygous, another is we call the they showing the truly homologous and unique. So it's not a pseudo. Sometimes you find a pseudo a SNP that will confusing you. We need to separate those pseudo SNPs from the real SNPs.
Okay, thank you.
In addition to this, we must have longer reads to identify these homologous SNPs or markers. It helps in identifying such SNPs by making comparisons of the sequence of surrounding regions. If this SNP is coming from blah blah chromosome or any other chromosome.
The ortholog, usually the SNP from the ortholog, that will be more meaningful because sometimes the homology, you will not sure whether that's unique or not.
Thank you. From Rahman, through gene editing, is it possible to increase long fiber percentage as well as gening outturn as a particular cotton germplasm?
Yes, I think that's another potential. I mentioned in Australia, the next technology advance of the gene editing will be should be used for this. There are lots of genes you can modify, and the fiber traits controlled directly or indirectly, involved directly, indirectly, about half of the genome. But some very critical elements you could identify and modify with genome editing, and that will trigger the fiber elongation and the second cell wall deposition to make the fiber stronger and longer. And like I mentioned, one case, even the susceptible to this gene, we could knock out with different editing, the silence tool. And before we use virus-induced gene silence, now we plan to use the gene editing. That's more effective, and also we will not. The USDA does not consider as transgenic advantages.
Okay, thank you. Now we are fastly running out of time. Dr. Kranthi, is there any question you can see that you think is the last question, the most important?
Yeah, that's a tough. Actually, there are at least 4 or 5 questions, which are good, but I'll let me take Dr. G. Balasubramani's question. He's asking, like, cotton fiber is more than 95% pure cellulose. Is it possible to increase fiber length and strength by overexpression of their associated genes? This is one, and associated is also one question from Dr. Sivaji Palve. He's asking, what is the role of cotton genomics in improving lint yields?
So this question for me or for
you? For you.
I think this question, I think I have already answered, I think, in Q&A. So yes, let first talk about the fiber length. That you know that if we let the cell wall to open for a longer period of time, to let more molecules, sugar molecule and others to enter into the elongated cells, then yes, we can. But again, you know, there should be some balance between all traits. If we increase fiber length, we may end up in losing some other traits as well. So yes, if I think a time will come when we will be able to do breeding at will, maybe after 5, 6, 7 years, when we will have the complete information of all genetic pathways which are involved in conferring such complex traits. As Dr. Yu said, around half of the genomes that is involved in shaping present day fiber. So how many genes we are going to target and which are the genes, this is yet to be determined at large. Yes, we know the information of very few important genes.
Yeah, thank you.
Yeah, yes, I just add one sentence. The ethylene pathway is one part of the system. The fiber is very complex traits and controlled by so many genes and the network. And obviously, we need to identify other major network and the pathway. And ethylene contribution regulation is one part of this.
Yeah, thank you very much. There is one last question. I mean, since we have, yeah, I mean, though we are running out of time, I'm still taking this liberty because it's so pointed. This is from Dr. Rajesh Patil from India. He's asking, on a scale of 1 to 10, where are we now in making molecular breeding successful in the near future? Now, this is for the two of you. It's very pointed because he's asking on a scale of 1 to 10, where are we now in making molecular breeding successful?
You go first, man.
Okay. I have answered this question, but again, I'm very optimistic, right? If we can integrate all approaches together, yes, then I will rate 10. If you are talking about only the science of genomics, how much it has the role, yes, it's around 10, 8, sorry, 8. Well, I'm quite optimistic. 8.
Yeah, I probably would be more conservative. If I rated the scale from 1 to 10, nowadays the potential of the cotton genomics, the power of cotton genomics, is far from the real reach yet, because as the human genome announced 2000 or 2001, people had a mixed reaction because in the 80s, when the project was launched, it was highly expected that the disease, the biomedical science will be revolutionized quickly. But like any new technology, that's the law of the technologies. Less lack. And for the molecular breeding, we had a couple of conferences before, and not just cotton, but other crops. The full potential of the cotton breeding has not been reached yet, and far from reached yet. So that does not mean the cotton genomics is not as powerful as before, as we thought, but it takes some time to mature, and the post-genomic application is our major goal. So at this point of time, I would rate probably 3 or 4, not just over 5. But the potential to reach 10 will be very, very possible in the future.
Good. Well, 2 very interesting answers there. And that's an ideal place to leave it. So a very big thank you to Dr. Mehboob and Dr. Yu for their presentations. From the questions, I can see that there's been a lot of interest generated, and the presentations will be on the ICAC website, and we'll look at if we can download the questions and answers which you've kindly answered, and we can then put that on the website as well. So next World Cotton Research Conference webinar will be on Wednesday, the 5th of May, where we will have another 2 internationally recognized speakers. With that, it's a very big thank you from me. Thank you to our interpreters, and we wish you health, keep safe, and we look forward to seeing you in May. And thank you to both our presenters for their presentations. Thank you.
Thank you.
Thank you, Kay, and thank you, everybody.
Thank you.
It was my
Thank you,
pleasure.
everyone.
Thank you, Dr. Rahman. Thank you, Dr. Yu.
Thank you,
so much.
Caroline.
Thank you, all participants.
Thank you, Chef.